the experiment that was done was the following. So we've started with some cells that are missing alpha-catenin. They're mutants that don't have alpha-catenin. Okay? They still have vinculin, they still have beta-catenin, they still have E-cadherin, but they have no alpha-catenin. And in those cells, alpha-catenin is missing from the membrane, of course, because it's not there. Vinculin, it turns out, is also missing from the membrane because it's just diffuse in the cytoplasm. Okay? So the protein is still there, but it's not localized anywhere. It's just forming a pool in the cytoplasm. Okay? And then we've taken these cells and added back alpha-catenin. So we knew already from biochemistry that alpha-catenin can bind to vinculin. So as an exercise, I'd like to ask, what can we conclude now from this experiment? Okay? And we're going to do this again with poll everywhere. Same story. Give me a 5 or 10 word answer and vote for your favorites. Let me just enable this. As I have to cooperate. It's very slow, sorry. Okay, I think this is okay. Are you able to enable it back there? You got it? Yeah. Okay, cool. All right, so discuss with your neighbors. Give me a 5 or 10 word conclusion from this experiment, and vote for your favorites. Also, a side note: the people who wrote this paper drew lines and arrows all over their images, so just ignore those and look at the cells. Okay. One other point to make: here in each column, I've labeled the protein that we're staining for in immunostaining, and then each row is an experiment. Okay. So these are all the same cells stained for 3 different markers. These are all the same cells stained for those 3 different markers. Okay, and I'll see if I can fix it. So the most popular response seems to be some variation of, you need alpha-catenin to get vinculin to the plasma membrane, right? Pretty straightforward. In the absence of alpha-catenin, vinculin stuck in the cytosol. When you add alpha-catenin, it can get to the plasma membrane. All right, so now let's make it a little more interesting. So we're going to do another experiment. We're going to add back alpha-catenin now in exactly the same cells, but we're also going to add a drug that inhibits the activity of non-muscle myosin 2. Okay? And you notice that now vinculin is no longer on the membrane. Okay? So, same story. Share with me, please, your conclusions from this experiment. Just activated it. Hopefully everybody sees it. Is it working? Yeah, okay, cool. And again, I'll circle around the room for questions. So what do we conclude from the fact that when myosin is inhibited, vinculin is no longer on the membrane? Okay, so the top vote getter right now is alpha-catenin and uninhibited myosin are both needed to recruit vinculin to the adherence junction. Okay, so that's great. That's an accurate description of the result. Okay, but so now what do we actually learn from that? Right. So let's take the interpretation to the next level beyond just describing the result. What does it mean that you need uninhibited myosin 2 to get vinculin to the junctions? That's what I'm after. Okay, so I have one response here that's kind of interesting. It says myosin is required to bring the vinculin molecule to the membrane to bind alpha-catenin. So that's a totally reasonable idea, except that myosin 2 is not a cargo-carrying myosin like myosin 5. Okay, so you guys talked about myosin 2 in muscles, right? What does it do? Pulls on actin and causes constriction, right? So when you inhibit myosin 2, you're not inhibiting the movement of vesicles or movement of cargoes within a cell so much as you're inhibiting tension. You're inhibiting pulling on the actomyosin cytoskeleton. Okay? Sorry?
It's closed, we can't change it.
Oops, I'm sorry. Which one? Yeah, I mean, I closed the one for experiment 2. We're going to do another one in a sec. Yeah. It's okay. You don't have to keep voting. No. All right. Okay. So you need. So the conclusion from this is that you need tension. You need pulling on actin filaments to get vinculin to cell-cell junctions. Right. So that's kind of strange, right? Why would that be the case? Well, let's take this experiment one step further and see if we can explain what's going on. So the next experiment that we're going to do is to start expressing some pieces of alpha-catenin, some fragments. All right. So let me give you one other piece of information before I show the data. So this is the domain structure of full-length alpha-catenin. We know that it binds beta-catenin at its N-terminus. That's what recruits it to the junctions. We know that it binds vinculin at this kind of intermediate region. Excuse me, and we know that it binds actin at the C-terminus. Okay? So we're going to express now a couple different fragments of alpha-catenin that are missing various amounts of the C-terminal domain. So they still have the vinculin binding domain, they still have the beta-catenin binding domain. Okay? So let's look at the results of expressing these constructs. So first, we're going to express this really short one, right? First 400 amino acids. And we're going to still inhibit myosin 2, just like we did here. Right? And the result is that short piece of alpha-catenin can recruit vinculin to the junctions, even though the full length cannot, when myosin is inhibited. Okay? So that's the first experiment. The second experiment is to express this longer construct. So now we're going to express this whole piece, and this is hard to see because this construct makes the cells a little bit sick, and it's hard to, they look kind of blubby and weird, but the result is that this larger construct, this larger fragment of alpha-catenin, cannot recruit vinculin to the junctions, even though we didn't inhibit myosin in these experiments. Okay? So when myosin is inhibited, full-length alpha-catenin can't recruit vinculin, but the fragment of alpha-catenin can. When myosin is active, full-length alpha-catenin can recruit vinculin to the junctions, but this fragment cannot. Okay? So this is a difficult one to experiment, but I believe in you guys. Talk with your neighbors, give it your best shot, and let's see what you come up with. All right, so I would love to give more time for this, but we're running short on time. So let me move on. Okay. So somebody nailed it. Somebody says a portion of alpha-catenin must be exposed by myosin pulling on it, and then vinculin can bind. Who was that? I'm just curious. Nobody wants to. Arturo, yeah, maybe. Nobody wants to own it. Okay, well, whoever it was, nicely done. That's exactly what the model is. Okay, so the key to understanding this experiment is to think about what's different between these two conditions, right? And what's different is this piece of the protein right here. Okay, so the strange result here is that the shorter piece of alpha-catenin can recruit vinculin, but the longer piece cannot. And so that indicates that this piece of the protein must be inhibitory in some way to its function. All right? And so we can suspect that perhaps this piece is somehow blocking vinculin binding. And it turns out we confirmed later using structural information that indeed this piece of the protein physically binds back and blocks the vinculin binding site. Okay? So then in order for vinculin to bind, it must be exposed by something. And what it's exposed by is force, by myosin pulling on F-actin, generating tension, and physically unfolding the protein. So here's that conclusion in cartoon format. So when there's no tension, alpha-catenin is all folded up and the vinculin binding site is blocked. When tension gets exerted by alpha-catenin, it exposes that binding site and allows vinculin to be recruited. Okay? So this was not an easy experiment to interpret. It took the field about 5 years to figure this out, and I gave you guys 3 minutes. So I'm impressed. You got quite a bit of the way there. The reason I wanted to go into that was to help you understand the kinds of experiments that people do, right? This is what modern cell biology looks like. Chopping up proteins, expressing pieces, using different drugs, and comparing different conditions to try to deduce how the cells work. Okay? So, yes, a couple of questions.
I think I missed cutting information, so why does vinculin matter so much?
That's a good question. It's almost a philosophical question, actually. Why does vinculin matter so much? So, you know, the assumption in the field is that you need vinculin because vinculin binds actin a lot more strongly than alpha-catenin. We think alpha-catenin binds to actin very weakly. And so vinculin coming on sort of cross-links and stabilizes that complex. That's the best idea that's out there in the field, but that's only an idea at this point. Nobody's really proven that. Yes?
I might have missed it. So you said that the short section is just missing the part that blocks it, basically. That's what you said, right?
That's right.
Okay.
That's right. Okay. All right. So that's what I wanted to say about the intracellular domain of coherence. So let's wrap up by just refreshing the key points. Okay. So I showed you cell adhesion matters in normal animal development. We talked about how coherins cluster on cell surfaces to bind to their neighboring cells and mediate strong adhesion. And then we talked about how they interact with cytoplasmic proteins and allow transduction of force between the actomyosin cytoskeleton and neighboring cells. And I didn't get to this last point, which is other types of cell-cell junctions. In the interest of not rushing through it, I'm going to leave you to review that on your own. The main point is just that there are other types of cell junctions as well. You should know what those types are and in a broad sense what they do, but you don't need to know those in so much detail. Okay? All right, so last thing, I would be grateful for your feedback on this lecture. I'm going to activate a poll everywhere question now. Your responses are anonymous, and I'll leave it open for the rest of the day. So if you have to rush off to another class, please take a few minutes later today to tell me what you thought. And I will be available after class for questions. I'll also put my email address back up. If anyone has questions about the material or just wants to say hi, feel free to drop me a note. All right, thank you.